Hi class, in this recording we're going to be focusing on cell junctions. When we think of a cell junction, I want you to think of something that connects two adjacent cells, something that will bind them together. And as we look at most cells in our body, they're going to be anchored to each other or to their extracellular matrix. As we look at cells, they use these cell junctions for communication with each other, to resist mechanical stresses, and to control what goes from one cell to another cell or what goes in between the cells. As we look at these cell junctions, we have a tight junction. This tight junction is the first kind of connection we're going to talk about. It's represented by cell adhesion proteins that are shown here in green. One protein will be anchored in the plasma membrane of cell A. The second protein is anchored in the plasma membrane of cell B, and they're almost like a zipper. These tight junctions are going to form very incredibly rigid, actually very tight, as their name implies, connections. They make it incredibly difficult for substances to pass in between cells. Generally speaking, when we think of tight junctions, I want you to think of them as a waterproof junction that keeps liquid from going in between the cells. The tight junctions are typically going to be against the apical end of that cell, so the end of the cell that's exposed to the cavity or opening. We also have desmosomes. A desmosome is like a tight junction, but a little bit more spread apart. So as we look at the desmosome, we're still going to have some cell adhesion proteins, but then there's extensions off of those cell adhesion proteins that will then be anchored into a intracellular plaque. And this intracellular plaque will keep those proteins anchored at the plasma membrane. These desmosomes are going to hold cells together in many ways like a clothing snap. It keeps cells from being pulled apart as a way of resisting mechanical stress. We also are going to have a Hemi Desmosome. A Hemi Desmosome is typically a half of a desmosome, and that half of a desmosome is going to be in the basal cells of a epithelium and anchor those basal cells to the underlying basement membrane. And this is what makes it very difficult to peel away epithelial tissues from the underlying connective tissues. We also have gap junctions. As we look at gap junctions, they're sometimes referred to as a communication junction because of this pore that's in the center, or that opening in the center of the gap junction. It has a ring of proteins called connexons, and these connexons are going to be arranged in a circle around a small opening that forms the pore. And this pore will allow for liquid to move in and out, for water to go through. And with the water, ions, nutrients, and other very small solutes will be carried with the water. We typically find a lot of gap junctions in cardiac and smooth muscle tissue and in embryonic tissues and the cells of our lens and cornea. Oop, I went too far there. Let me back up. Two slides ago I had mentioned hemidesmosomes, but I didn't have a picture of it. Here are the hemidesmosomes. So it's half of a desmosome that's anchoring the cell to the underlying basement membrane. And we can contrast that hemidesmosome with a desmosome, which is right here, that is a cell-to-cell connection. If you have any questions or comments about cell junctions, please let me know by posting in the class discussion board or by shooting me an email. And as always, happy studies.